Okay. All right. Okay, we got started here. We still got 8 and I haven't seen anybody else join, but I'm gonna try to share the screen again so we can, there's no good, well, let me go ahead and say that this topic, there were two videos, one that had 41 people view it and one that had 25 people view it. And I painfully watched myself day before yesterday for one, wasn't so bad. And then the second one was, and they were both long, but the second one was just, I mean, I put myself to sleep. But it's not that the topic wasn't good, it's just that I didn't, for some reason, didn't have I don't know, I just was one of my, Steve, I noted to Steve, it was one of my worst times. And he said, even good guys have bad times. But bottom line is that the material was good, but I just didn't seem to have the good delivery. Now, there are some handouts. Let me, well, I'm going to have to share the screen to do it. Well, let me go ahead and say this, that there is in botany no more, it's not, there's some topics that are extremely difficult. Plant reproduction is one of them, especially when you start talking about angiosperm reproduction. Another topic that's difficult is the photosynthesis and respiration part of plant physiology. In other words, the biochemistry. They're difficult. And I know of hardly any subjects in botany that are more difficult than that. So, and you think about, well, what possible good could those topics, being so esoteric and so hard to master, what possible good could they be? Well, recently, and Donna knows this because she watched it too, on the PBS, they had the chemical, they have a program on I guess it's NOVA, and it has to do with element chemistry. And anyway, they dealt with photosynthesis this week, and they brought up a good point. They talked about something I didn't talk about in when I did this photosynthesis part of physiology in 2021. They talked about, you could write this down if you want to, capital R and then U, capital B, I-S-C-O, rubisco. R-U-B-I-S-C-O. The R, B, and C are capitalized, and that is an acronym for a protein, actually a protein that's an enzyme. It's called ribulose biphosphate, actually ribulose 1,5-biphosphate carboxylase oxygenase. Now you can know why they shorthand it, because it's such a long expression. Typical enzymes are proteins, and that protein is the most abundant protein on the face of the earth, they say. And what it is a 5-carbon compound that enzyme that catalyzes the reaction of take a fixation of carbon dioxide to ribulose phosphate. Ribulose phosphate is a 5-carbon compound, and you take here. Let me put this up to illustrate that. Oh yeah, I'm not on screen share, so what good is that? Okay, all right, screen share. All right, now, can you see that? Can y'all see that diagram?
Yeah, we can see it.
Well, what it is there's the ribulose biphosphate, and it's not showing ribulose rubisco, the enzyme that catalyzes the addition of carbon dioxide to the ribulose biphosphate, is not showing, but it's right about here is where it would occur. Can you see me moving my arrow? Yeah.
Yes.
Okay. I'm not going to mess with that annotate thing. It's just not worth it because the arrows. Anyway, you add a 1 carbon to a 5-carbon compound, and you make a 6-carbon compound, it immediately breaks up into 2 3-carbon compounds called phosphoglyceric acid, which converts to glutaraldehyde 3-phosphate. And 2 of those go together to make a 6-carbon compound called glucose, or sugar, the basic building block sugar of coming out of photosynthesis. And so the reason I mentioned that, and I didn't know this until the other night, they said, and I knew photosynthesis was supposedly not very efficient, and they said part of the problem, the reason it wasn't very efficient, was that ribulose biphosphate, Rubisco will actually, instead of picking up carbon dioxide, gets competed. The carbon site gets competed with by oxygen. So instead of picking up a CO2, it'll pick up an O2, and that means that that makes the process inefficient. That photosynthesis is not fixing the carbon it could fix, because the enzyme that ordinarily would catalyze the reaction of RUBP with CO2 is being short-circuited by the competition for the site on the enzyme for oxygen, with oxygen. So the people in Illinois and the university were working on basic, trying to fix that, trying to figure out a way. And basically what this has to do is feeding the world's people. But my point is something seemingly so esoteric that nobody would really want to learn it for any good reason, has a good reason to learn it. So, so that's the point of all I'm getting at here. And then the same could be said. The same could be said with respect to reproduction. Okay. Studying reproduction could have some ramifications that would go a long way too. Let's see if I can. All right, I'm going to go down and pick up, and while I'm on that subject, there's about 32 slides associated with plant reproduction with all of the ads that I gave you. Now, I guess no one else has come in, Steve, but
we had one more come in right after you first started.
Okay, good. Well, this, okay. Oh gosh, why did it do that? Oh, great Scott. For some reason, it picked. I'm trying to. The PowerPoint's not working well with the screen share, and I don't know why. Oh, get that out of there, jeez. All right, let me go down here and pick this up. Let's see if I can get rid of, I think I'm gonna see if I can minimize that. Okay. All right, the very last slide that would be part of the reproduction process. This slide, these 4 slides here are intermediates between plant reproduction and genetics. And tomorrow or today or tomorrow, I'm going to put up the genetics material Roman here. But these 4 slides should be that one should be the leadoff slides but what I'm getting at is this alga, this simple alga here, Chlamydomonas, that I spent some time talking about. It is the organism that Rubisco study was taking place with. They've picked a simple alga like Chlamydomonas that has a 1-celled adult, seen in the center here, to be the study organism for that photosynthesis process I just talked about. So that was I thought that was interesting. That wasn't on the program, Donna, but I did learn about it. So, and this thing is just not written. Something is, okay, I cannot. Okay, I had to hit escape a million times to get that, Steve. I don't know what's going on, but I can't blame the devil from hell, I don't think. All right, now this slide is the intro slide. I guess I'm gonna run through. Has anybody got any immediate questions before I kind of run through these slides really quickly? Russell asked a good question, and I answered it in an email, but didn't send it to everybody. But this this is the intro slide on plant reproduction with the asexual reproduction being compared to sexual. But the hallmarks here really are meiosis and fertilization. Fertilization is a synonym for oogamy. Oogamy, oogamy, is one of the three kinds of gametic union known as syngamy, the most evolved. The kind that fertilization or oogamy is when there's a large non-motile egg and a small motile sperm. And in the case of angiosperms, there's no flagellum, it's motile through a pollen tube. And that's true of conifers too, gymnosperms too, well, some gymnosperms. Now, this just emphasizes asexual reproduction. That gives a lot of numbers real quick, but there's no disadvantages, there's no variation like genetic sexual reproduction will do. This is the same thing going on with quaking aspen. Now, this is I think a very good slide because it really sums up alternation generations, which is what our aim was. Now this slide here, this one at the bottom left is alternation of generations. And this is just a generic look at it, emphasizing fertilization and meiosis and as the hallmarks and everything revolving around them. And despite my difficulty in presenting it in session 2, it still always amazes me that this very thing is something that people just can't seem to grasp or get. They think you got to memorize stuff. Really, if you understand what fertilization is and what meiosis is, and meiosis is difficult to comprehend, if you understand those two and where they fit, everything else falls into place. And also it's important to understand the difference in a gamete and a spore. And that's, spore is a cell that grows into an adult, where it's a single cell unicellular chlamydomonas cell or a filament or a moss thallus. By the way, I did send you answers to those exercises. So let's see. All right, everything revolves around syngamy and meiosis. And I spent too much time on both presentations on this slide, but trying to focus first on the part that you have, the upper left. And chlamydomonas has this one on the upper right, and plants and higher algae, some of them have the bottom left, the alternation generations. And sometimes it's predominantly diploid with a little haploid like this fern, and the fern allies was that way too. And sometimes, like in the case of the moss, it went backwards with the haploid generation prevailing over the diploid generation, and the sporophyte, the 2N part, being parasitic on the gametophyte. And in this case, water splashes from down into the neck cells dissolve, and the sperm goes down here and fertilizes the egg, and that's where you get your zygote. And then, of course, the zygote divides by mitosis and forms an embryo that's not shown, and you get this sporophyte, which is got a foot embedded here in the gametophyte and that parasitizes off of it, the seed in the capsule. And in the capsule, there's spore mother cells that undergo meiosis, and that's what's going on here. And the spores germinate down here. And the GM would stand for germinate, but it should, because it's not a gamete, it's a spore. So this is prevalently haploid. This is a homophorous life cycle, like most ferns and fern allies and most all mosses. And there's only one kind of spore in the cycle, and only grows into one kind of gametophyte, like this one. This one here. Okay. Then this is the heterospous life cycle that's typical of higher plants and a fernali called Thelaginella, spike moss. And there's two kinds of spores, and they produce a gametophyte that bears eggs and a gametophyte that bears sperm. And if you're heterosporous, two kinds of spores, you're automatically going to be heterophthalic, have two kinds of gametophytes. And then they unite to form the zygote, the first cell of diploid generation, and then embryo and the sporophyte. And the sporophyte produces two kinds of sporangia, one bearing megaspores and one bearing microspores, and they undergo meiosis. These sporocytes is another name for spore mother cells. And so now this is the slide that I referred to as the Holy Grail slide, because it's I've never seen a slide that had as much information in one slide as this one, because it is just it brings together the plant kingdom plus algae if you don't put algae in the plant kingdom in terms of the terms. And then the embryo sac, by the time we get to flower plants, is gone. I mean, the archegonium, by the time we get higher plants, is gone, and we get something, an 8-cell entity called an embryo sac. And actually, it is this called that because it eventually houses the embryo. And the embryo sac is a synonym for the megagametophyte, and the pollen grain is a synonym for the microgametophyte. And that's what the answer to that question is here. What are these cells on the previous slide? If you can understand that, that really helps. So I first came across this slide in a lab manual in 1972. And it's just something that's been useful ever since. I never saw it anywhere else. Now, this is a generic look, an abstract but generic look at alternation generations. And the way this diagram is going, it representing equal time, which is not necessarily the case, depending on where you're dealing with ferns or mosses or higher plants. This is a cartoon version and a more accurate drawing version on the right of three kinds of syngamy. The primitive type is the gametes look identical, it's called isogamy, but they're chemically different. Two pluses will do nothing in a Petri plate, two minuses will do nothing, but you put pluses and minuses, and running bear will meet little white dove in the middle of the Petri plate. And they'll form a line of zygotes, whether it's algae or fungi. And that brings me to a point. One of the reasons it's difficult for sessions like this is we don't have a laboratory. We don't have people looking under the microscope and having hands-on with live organisms. Then this less common type, where the female is still bigger than the male, like the ultimate down here, but they're both motile, and that's called not isogamy or anisogamy. And then oogamy is the type that you are most familiar with. In the human, of course, the sperm are not anterior. I mean, the flagella are not anterior. There's one posterior one instead of two anterior ones. This anterior one is really what you'd get in some algae. And all right, this is where there was an error that I made, and it was an error sheet that I can't remember for sure if I spelled it out, but I actually said it is, in video, I said it is, mitosis is thought of as a sexual process, but the slide clearly says it's not. And that's correct, it's not. Mitosis can occur in any cell, where it's haploid or diploid. But meiosis is the most difficult concept to get, probably, in all of the stuff we have. And I think that item 11 on that errata sheet, it's not totally an errata sheet, actually has a definition of meiosis. And I could rattle it off now, but it would just, it's a very complicated, meiosis is two nuclear divisions. The first is a reduction division, where you separate the homologs, and the second is a mitotic division, where you separate chromatids. note the similarity between this here and this up top, because the second part of meiosis is mitosis. That's why there's a similarity here between these four, the two up here and the two down here. So, with any questions about this? Okay, the only thing wrong with it is you don't really see the chromosomes during interphase. When you look at interphase cells that are not dividing, which is 90% of the time, you see a nucleus that has a bunch of dots. And if it's a sectional view, you see a bunch of chromatin dots and that run in front of the screen and behind the screen. You have to think 3D when you're looking at it, because you're looking at a thin slice of, I mean, can you imagine what a thin slice of a piece of spaghetti would look like? Speck, really. And that's what, so the chromosomes are not short, thick, and contracted when they're replicating. They're long strands of chromatin granules. This by itself is so complicated it's worthless, but the hallmarks are still there, meiosis and syngamy, or meiosis in this case, fertilization. And here it begins to get a little more intelligible, with the emphasis being on the last diploid cell in the sporophyte generation, the megaspore mother cell and the microspore mother cell, also known as sporocytes. And when they undergo meiosis, you wind up with the first cell of the haploid generation. Here, the tetrad, all four survive. Unlike the human female, only one of the products of meiosis survives. That's an interesting counterpart between flowering plants and humans. That the products of meiosis, 3 of them die out in the female, and only one survives, the one furthest from the micropyle. If we were actually doing a lab, we'd be looking at slides like this one at the bottom right, and actually could see this, go all of these detail, or see this pollen grain over here in detail. This is, well, microsporogenesis and microgametogenesis are fancy terms for generation of spores and generation of gametes. But just really, this is meiosis, and then this is what happens when the spores germinate to form a germinating pollen grain. Gets ultimately 3 cells or nuclei, 2 sperm and a tube nucleus. And at first they're 2 cell, the generative nucleus generates sperm. And the more complicated female has an 8 cell instead of a 3 cell gametophyte. And this wall here is a megasporangial wall, and everything inside of it is haploid, but the wall itself is diploid, and it's not labeled here either. Everything gets labeled. Actually, nucellus, I'm glad I saw that. Nucellus is a synonym for the megasporangial wall. That's an important oversight on my part. Anyway, all right. This is the details of all of that going on, and there's the egg in here, and the micropyle is here, and there's the megasporangial wall or nucellus. Okay. All right. That's just a comparison of haploid and diploid. This, when I mentioned and answered Russell's question about the very cryptic gametophytes of higher plants, this is what I was talking about. The microgametophyte or the pollen grain, or the megagametophyte with 8 cells, microscopic. In humans, the haploid generation is very cryptic too. Sperm and eggs are pretty microscopic, pretty hard to see. So as we go through evolution, the sporophyte becomes prevalent and the gametophyte becomes not prevalent. And those are in the beginning of one of the videos I told you to add these. Well, they were already added, slide you got. But when I taught this originally, and Steve was part of this, and actually Donna was too, I asked them to add these. And that next slide is details of what's going on in pine. And all this is haploid nuclei, the old female megagametophyte. And this doesn't look like an archegonium. It doesn't look like the vase that we associated with ferns and mosses, but it is in effect essentially an archegonium with an egg down here. And the egg gets fertilized and forms a zygote, and the embryo starts developing and starts feeding off of this haploid tissue. One of these sperm just withers away, and the other sperm goes and fertilizes the egg. Takes a year in pine to go to growth after it enters the micropyle. It takes a year to grow through this megasporangial wall, or the nucellus wall. And then, so you see a big pine cone, that's 2 years old. The first year, it's only 1 centimeter or an inch at most in diameter on a typical pine cone. Now, this looks like a dicot because it's got 2 cotyledons. It's pine, but what you're looking at is a sectional view. This is a part of a cotyledon that got cut away in the slide making. In other words, you see these? Pine, unlike dicots, is polycotyledonous. Whereas there's one of these cotyledons, there's another, but this represents one of the others and all the rest of the cotyledons weren't a part of the slice, weren't part of the cut. So anyway, this is an embryo of pine. Okay, what the process is called, the whole thing, what is the pleuroty of the tissue around the embryo? It's megagametophyte, and its function is to feed the embryo. And it's called the megagametophyte tissue. This is another look at this. Another way of looking at that other slide showing the reduction in the gametophyte generation and evolution. This is just a summary slide. And okay, this is just emphasizing that actually it's emphasizing an isomorphic alternation generations where the sporophyte here looks identical to the gametophytes. And the adult in this algal filaments, the adult is a filament, whereas the adult in this is a membrane, and the adult here is one cell. So the adult here is a giant kelp 300 feet long, and these little microscopic gametophytes are the adult haploid generation. So this is isomorphic, a repeat of the isomorphic, heteromorphic, like this cycle here is like bryophytes and ferns. And it's more like ferns because the sporophyte dominates, unlike mosses where the haploid dominates. But what does what does a kelp, like a 300-foot long kelp off the coast of California, have in common with a moss or fern? It's heteromorphic, alternation of generations. Okay, isomorphic alternation of generations when they're like that. And then, of course, this, I always love this slide because it's so simple, it's complicated. And the fact that if I ask the question, how do you tell zoa mitospores from gametes? And it has to do with where they come from and what they do. These come from meiosis, these come from mitosis. And what do spores do? Spores don't fuse, they grow into adults. Gametes, on the other hand, their fate is either one of two things. They either die or they unite. And okay, these are intro slides that'll be covered in the genetics ones. Okay, that's a little bit different approach to what I did in most of the question and answer sections, but I hope I went faster than I did when I did it in 2 sessions. But now this thing's working fine now, Steve. I hit escape and it works. Go figure. So But all right. I hope that was good for you. I hope that helped. Anybody, questions? I hope that you got something out of that. Anyhow, all right, so today or tomorrow, I'll put up the genetics, first of the genetics, and it'll lead off with those 4 slides that bridge the gap between reproduction and genetics. And as we noted, there is no evolutionary class in this subclass in this botany, but when you do evolution, you study evolution, you have 2 major components. It's genetics and ecology. And I talk a little bit of ecology, but I don't break it out separate like I do genetics. But anyway, all right. So glad y'all are still sticking with me. Apparently, at least 20-something people are still with us. Because that's about how many had looked at this second one. But I told Steve when I looked at the second one, I saw that 41 people had looked at the first plant reproduction. That means one of 2 things. It means some people looked at it twice, since there's 28 people in here, or some people not even a part of the class are looking at it. And that's, since it's public, that could be.
This is Carolyn. I frequently look at them a second time.
Well, when it's, Carolyn, thank you. When it's that long, you have to. I tell you, I thought to myself, anything over an hour, even the best minds tend to lose it after that. So especially something as tedious as this. But anyway, keep in mind that I'm not bragging, but what we just went over, there's probably only a handful of schools in the whole country that would even cover that topic. And Congress, a lot of these Congress people we have, don't understand why you do basic biology, because what possible good could it be? Well, what I just said about the way to improve the efficiency of photosynthesis would be a good example of that. And sometimes applied answers to applied science come out accidentally when you do basic science. So anyway, I mean, studying reproduction could help you solve problems of exotic plant reproduction. So, and I'll tell you, most of the exotic plant problems are plants that do asexual reproduction and sexual reproduction. And that's what makes them really bad plants. I've got a plant out there in the yard that's related to gladiolus, and it produces asexually by corms, and it's just a nightmare. So I planted it because it was pretty, and that was a mistake. So, but anyway, all right, good to see y'all. All right, take care.
May I ask you an unrelated question?
Please, Span, go ahead.
Tupilaria, when does it bloom?
What's that, Tupilaria?
Tupilaria. I mean, there's a bunch of them in the woods behind my house, but I've never seen them bloom.
Well, I honestly don't know off the top of my head without looking. I mean, I can look right here. I'm sitting here. If you'll hang on. Yeah.
Okay, so the Tupilaria, they just get their energy now and put it into the roots from the leaf. While they can get the energy from, because the leaves aren't on the trees yet. So the leaves get their energy, they wither away and die. The Tupilaria bloom, the orchid comes up in late summer, early fall.
Oh,
Okay.
Okay, that's what I wanted to know myself, and I couldn't remember.
And without any leaves, so there are no leaves up when you get the orchid stalk.
okay, got it. Thank you.
Okay, it's interesting when I look in the Florida book on Tupilaria, it says summer. So, but anyway, it depends on. That's one of some liliaceous plants. Lily family has that too. I've got something in my yard that's not native called September lilies, black chorus, beautiful thing, but it flowers and leaves at different times.
Right.
Yeah, I'm familiar with those. Yeah,
I like them. Even though they're not native, they stay in my yard. I've never seen them get out of a yard. I've yet to figure out how they get around something, some mole or shrew or something must carry them around because they don't reproduce sexually, I don't think.
Oh,
But the little corms or whatever they've got, the bulbs keep come up anywhere and you never quite know where. And I like them because August doesn't have, I don't have a lot blooming in August,
mhm.
September. So that they take over. So, I mean, they don't take over the yard, but they fill that role. So, okay. That was a good.
Thank you.
All right.
Thank you.
Thanks.
Bye.
Thanks, Bobby.
Thank you.
Charlie, where do you live? I forgot.
Thank you.
I live in Marietta.
Okay, well, Kevin Doyle, our newsletter editor, lives there too. Of course, quite a few people do, I think, in our society.
Yeah, a few million live around here, yeah.
Yeah, all right, bye.
Right. Well, thank you now, appreciate it.
Okay.